Barts Health provides services for around 2.5 million people in East London, and there are 5 separate hospitals within the Trust. The majority of the lab tests across the Trust are carried out in the Pathology and Pharmacy building, which is at the Royal London Hospital in Whitechapel. The people who work in the diagnostic laboratories include medical doctors, scientists and support staff, and they work closely with the healthcare teams caring for the patients in the hospitals and the community. In this video presentation, we will look at some of the laboratory diagnostics and will base them around a fictitious patient who we'll call Fred Bloggs. Fred is suspected of having the hematological malignancy acute myeloid leukemia, or AML. We'll start off the video by looking at 2 areas that are essential in making the diagnosis. These are immunophenotyping and molecular cytogenetics. One of the treatment options that may be considered for our AML patient is a stem cell transplant. A key feature in this procedure is identifying a suitable stem cell donor and HLA matching the donor to the recipient. We'll look at how this work is done for patient Fred in the clinical transplantation laboratory. Whether the AML patient is treated with chemotherapy or a stem cell transplant, the treatment leaves the patient immunosuppressed and susceptible to a range of infections. We'll look at one such infection in the video, cytomegalovirus or CMV, and how CMV viral load testing is used to identify infection and monitor antiviral treatment. We'll also look at how other infections are diagnosed in the microbiology laboratory. So we'll start with a tour with immunophenotyping.
The Immunophenotyping Laboratory is part of the Specialist Integrated Hematological Malignancy Diagnostics Service framework. We are a small laboratory that performs immunophenotyping assessments of patients with suspected hematological malignancy using a process termed as flow cytometry. The samples are received into the Pathology building and the Specialist Integrated Hematological Malignancy Diagnostics Service reception. They're recorded in the daybook and then booked onto the Hospital Laboratory Information Management System. All samples are unbagged, checked, the demographics are checked against the request form, and then written into the manual daybook. The samples are then booked onto the Hospital Laboratory Information Server using its very own unique patient identifier. All immunophenotyping results have to be interpreted in an integrated manner with morphology. So here we are making peripheral blood smears of our patient's sample. These slides are then stained in the hematology lab using a May-Grunwald-Giemsa stain. Slides are loaded onto an SP1000 automated slide stainer. They're stained using the May-Grunwald-Giemsa stain, and you can see here that the slide got a blue background. We have labels for our patient. We have a T cell tube, a B cell tube, and a myeloid tube. Each individual label is stuck to a falcon tube, which is a plastic tube that can go on the flow cytometer. The sample is inverted to ensure that the cells are mixed correctly, and the appropriate volume of blood or bone marrow is then aspirated and placed into the bottom of the falcon tube. We're now adding an ammonium chloride based lysing solution into each of the tubes. This lysing solution is there to obviously lyse the red blood cells and their precursors. Here we have the tube. You can see the red cells have lysed over a period of time, usually about 10 minutes. You can see that it's quite transparent and you can see through the tube. So we now fill the tubes with a phosphate buffered saline solution, which is optimized for flow cytometry. It contains sodium azide. These are loaded into the centrifuge and spun roughly between 200 and 400 G. Once the centrifugation step is finished, tubes are removed from the centrifuge and the white cells have obviously been pelleted and are stuck to the bottom. So the supernatant is tipped out and poured down a sink, ensuring that the white cells are stuck to the bottom of the falcon tube, and then some water is flushed down the sink just to remove any biohazard contamination. The next step is to add the antibodies, which are going to target our proteins on the surface of the white blood cells. So these antibodies are kept at 4 degrees. They're also kept in dark bottles we have a cocktail of antibodies targeting the T cells, one for the B cells and one for the myeloid cells. These cells are then mixed. Once the cells have been mixed and the antibody is bound, then incubated, they're then ready for analysis on the flow cytometer. The cells are loaded onto the sample injection tube and held with the aspirator arm, and then acquired through the flow cytometer. And here you can see the lights flashing. For each light, a single cell is counted. The tube is once then removed following the acquisition of the sample. So this is the printout that we get from the flow cytometer. Here's our patient, Fred Bloggs. This is his first tube, tube 1, which is the T cell tube. As we said, everything is gated against 45 versus granularity, the side scatter. This is a forward scatter versus side scatter, size and granularity plot. Gate on the 45, exclude all the debris down here. Gate on our lymphocytes, which are falling here. And we look at CD3, CD4, CD8, CD57, 56, and CD10. So you've got CD4 versus CD8. Here's your normal 4s and 8s. So you've got your 4s and 8s roughly add up to your CD3. Your CD8 positive, 57 positive large granular lymphocytes, your natural killer cells. And then we go to something called P1, which is this region down here, this blast region, which has very low side scatter and dim 45. And we colour those in blue, and we can show them on these plots here that they're negative for 4 and 8, negative for 3 and 56, negative for 57 and 8, and negative for CD19 and 10. And these are the percentage values of the expression within that P1 population. Here we have our second tube, our B cell tube. Again, we're looking at CD45 versus side scatter, gate around the CD45 region, our lymphocytes and our proposed blasts, got size versus granularity. On this axis we have CD19, our B cell marker, and CD5 a T cell marker. These are our residual normal B cells, these are the residual normal T cells. If we look at our lymphoid region, 26% of the lymphocytes express CD19 with a nice even distribution of immunoglobulin light chain, kappa to lambda, falling here and here in a ratio of just over 1 to 1. If we get on P1, our blast cells, we colour code those in red, we can see that they're negative for 5 and 19 and negative for 23. This is reflected in these percentage values here. This is our tube 3 of our screening tube, our myeloid screening tube for Fred Bloggs. Again, we have 45 versus side scatter, exclude the debris, gate around all the white cells. Here we're only gating on P1, our blast cell population of interest. This is where they fall in relation to size and granularity. If you look at this axis here, we've got CD34, which is a transmembrane phosphoglycoprotein, which has been identified on hematopoietic stem cells. We've also got on this axis CD117, that's a proto-oncogene known as c-KIT. It's a protein tyrosine kinase, and it's found on multipotent stem cells, hematopoietic stem cells, and also the common myeloid progenitor cells within the bone marrow. And you can see that this P1 population is coming out for both positive for CD34 and strongly positive for CD117. If we look at CD34 along this axis versus HLA-DR, CD33 and 13, you've got a sort of bimodal but positive expression of HLA-DR, a sort of moderate expression of CD33, and a moderate to strong expression of CD13. There's no CD14 because the blasts are falling in the negative region, so they're not expressing any sort of monocytic markers, no 11B. If we look at the percentage of expression on this blast cell, we can see that 13, 34, 117, HLA-DR, and CD33 are all coming up strongly positive, and our monocytic markers, CD14 and CD11B, are coming up negative. So this is the last tube I'm going to show you, or the ALOT tube, or the acute leukemia orientation tube for our patient. Again, we're only interested in this blast population. Gate around CD45, exclude anything that's 45 negative. Within this tube, we're looking at cytoplasmic CD3 on this axis versus surface CD3 on this axis. So we're looking for our T cell markers, and you can see here that our blast cells are falling negative for both cytoplasmic and for surface CD3. If we look at this axis, we've got CD79A along the bottom here. We've got myeloperoxidase tagged to a FTSE fluorochrome on this axis. And you can see here we've got very strong expression of myeloperoxidase on our blast population, but no evidence of cytoplasmic CD79A. This axis is CD19 versus myeloperoxidase, and you can see here we've got nothing falling within our CD19 B cell gate. Everything is falling up in our myeloperoxidase. This axis shows CD7 versus myeloperoxidase, and there may be a very small amount of CD7 expression, very weak expression possibly on this progenitor population, but it's very unlikely. It's just these blast cells appear to be quite granular in that sense. If we look at our blast cell population here, we've got cytoplasmic CD3, CD and surface CD3 are both negative, myeloperoxidase very strongly positive, no 79A, a little weak bit of CD7, but it's less than 20%, so we're not calling that a negative, no B cell, and CD34, which is coming up down here, is positive as we saw before. So we can say clearly that this case is acute myeloid leukemia. It's not mixed phenotype because it doesn't express CD19 and doesn't have expression of cytoplasmic CD3. So it's a clear barn door acute myeloid leukemia. Once the immunophenotypic analysis has been complete, the slides and the results are then reviewed at the multidisciplinary team meeting. And here we are looking down the microscope. This is at ×40 as we're scouting around. Certainly an absence of platelets, thrombocytopenia. The patient looks anemic, and you can see that there's a rather homogeneous population of white cells floating around that have a high nuclear to cytoplasmic ratio. This is very common of blast cells. And as we go down to a lower power, looking at ×10, to get a bit more of a global assessment, you can see there are quite a few of these blast-like cells down the microscope. So the blast cells that we're seeing down here and the immunophenotypic results suggest that our patient has acute myeloid leukemia.
Welcome to the cytogenetics laboratory. We study chromosomes to look for structural and numerical abnormalities. These abnormalities have diagnostic and prognostic significance in a number of cancers. Here we see the bone marrow sample received from Fred Bloggs. The sample is assessed by a scientist prior to processing to check the reason for referral and ensure the correct culturing protocols are used. The labelling of tubes and paperwork is also checked to minimise any mistakes. We see Lorena setting up cultures from Fred's bone marrow sample. To visualise chromosomes, we need dividing cells. Chromosomes are only visible when the DNA is at its most condensed. This is during mitosis. The cell cycle of a lymphocyte lasts 24 hours, and mitosis takes about 40 minutes, so we have a narrow window of opportunity to view the chromosomes. We therefore need to manipulate the cell cycle to ensure the cells are harvested at the right time. Lorena adds cosamid, a mitotic spindle inhibitor, to the culture, which stops the cells passing from metaphase to anaphase. This greatly enriches the population of metaphase cells. The culture is then incubated overnight at 37 degrees Celsius. The following day, the cells are harvested. This process starts with a centrifugation step to remove the medium. The cells are then treated with potassium chloride, a hypertonic solution causing the white blood cells to swell and the red blood cells to lyse. After 15 minutes, the cells are pelleted once again by centrifugation. The cells are now preserved in their swollen state by fixation using a 3:1 methanol to acetic acid fixative. This fixative also neutralizes any infectious organisms and breaks down the lysed red cells. It is important that this first fix is added drop by drop to prevent cell clumping. This fixation step is repeated twice more until a clean pellet of white cells is visible in the tube. The fixed white blood cells are ready to be dropped onto slides. This is done from a height. As the cell membranes are hardened from the fix, they shatter as the cells hit the slide. The chromosomes spill out from the cells and spread onto the slide. These slides can be utilised for a variety of procedures, including G-banding and fish. G-banding involves pretreatment with trypsin, a proteolytic enzyme followed by staining with Giemsa or Leishman stain to create characteristic light and dark bands. The slides are mounted with glass cover slips and are ready for analysis. Not all samples yield chromosomes. In these cases, we use fluorescence in situ hybridization, FISH, to address specific, focused clinical questions. Fluorescently labelled DNA probes are pipetted directly onto the slides with the patient cells. The probe is then covered by a glass cover slip and sealed with rubber cement. The probe and target DNA are denatured at 72 degrees for 2 minutes to give single-stranded DNA. The slide is hybridized overnight in a humidity chamber at 37 degrees and the complementary sequences anneal. The next morning, the slide is washed, removing unbound probe. FISH can be applied to metaphase chromosome preparations and/or interphase cell nuclei. FISH is used to assess the presence, absence, or the copy number of specific DNA segments by fluorescence microscopy. Here we can see Amy analysing the chromosome from Fred Bloch's bone marrow. The chromosomes are viewed using a light microscope, and all 46 chromosomes are examined and compared as homologous pairs. Fred has an abnormal karyotype with 47 chromosomes. 3 copies of chromosome 8 have been detected, referred to as trisomy 8. In AML, cytogenetics is considered to be one of the most important independent prognostic indicators. Trisomy 8 is a recognised finding in AML and is classified as an intermediate risk abnormality.
When a patient is being considered for stem cell transplant, we ask the clinical teams to send blood samples from the patient and any siblings that may be possible donors. Often the siblings live overseas, so we receive samples from all over the world. The nurses send blood tubes out to the siblings living abroad and they courier them back to us. In the case of Fred Bloch's, we received his samples and those of his 2 siblings, one living in New York and the other in Jamaica. This is where the samples are received and booked into our database. The first step in the testing process is the extraction of DNA. This used to be done manually, but is now done using these small robots. These robots can extract DNA from up to 6 samples at a time. All of the reagents required for DNA extraction are present in these small cartridges. The cartridges are loaded onto the robots, followed by the blood samples. Importantly, the samples are checked before the extraction run starts. An extraction run takes approximately 20 minutes and yields 300 ml of DNA at between 50 and 100 nanograms per microliter. Our first typing technique is PCR SSO, also known as Luminex typing. This method is relatively high throughput and gives a medium resolution HLA typing result. We type for HLA class 1 and 2. The technique starts with a PCR in our PCR setup lab and requires DNA, primers, tack and buffer to be added to the plate. The plate is then taken through to our post-PCR laboratory where it is loaded onto a thermal cycler which runs through the cycling parameters to amplify the DNA. Next, we need to visualize the amplified DNA. The PCR products are loaded onto an agarose gel for size separation by electrophoresis. We can visualize the amplified DNA by looking at the gel with UV light. In this case, you can see that our samples have amplified, including those of Fred Bloch's and his siblings. These post-PCR products are then mixed with beads, which have a surface chemistry that allows them to be chemically coated with a number of different targets, including short HLA oligonucleotide sequences. Complementary strands of amplified DNA bind to the oligos on the beads, and unbound PCR products can then be washed away. We call this the hybridization stage. Following hybridization, the bound products are labeled with fluorochromes so they can be visualized. The whole process takes approximately 2.5 hours. The samples are then ready to be run through the Luminex machine. The Luminex machine is essentially a modified flow cytometer. It has 2 lasers. One excites the fluorochrome in the center of the bead, and the other excites the fluorochrome attached to the bound PCR product. Once the run is finished, the data is analyzed in the office here. We can see a pattern of positive bead reactions, which we interpret into a medium resolution HLA type. Fred Bloggs' sister in New York is matched at medium resolution and is therefore taken through for high resolution typing by Sanger sequencing. So we go back to the original DNA tube and back to the PCR setup lab. Sanger sequencing is very time consuming and detailed work with a lower throughput compared to Luminex. So only siblings or related donors that are matched at medium resolution would continue on to high resolution. In this case, Fred and his matched sister will be sequenced. We start with an initial PCR, which is a generic amplification of each HLA locus. Reaction mixes are set up in thin-walled PCR tubes. The PCR tubes are then taken through to the post-PCR laboratory. Similar to Luminex, the PCR is run on thermal cycler and the products are electrophoresed on an agarose gel to check for amplification. Once amplification is confirmed, the cycle sequencing reaction can be set up. Each sample is set up with a forward and reverse reaction for each exon of interest. For example, for each HLA-A locus to be sequenced, exon 2 has a forward and reverse reaction set up, exon 3 a forward and reverse reaction, and exon 4 a forward and reverse reaction. The reaction plate is firmly sealed and then placed on a thermal cycler for cycle sequencing. This is the sequencer the samples are run on. It is a shared piece of equipment between departments. It has 96 capillaries, which means 96 reactions can be run at the same time. Plates can also be stacked up here. Back in the office, the sequences for each run are then put together and analysed. Our data showed that Fred and his sister are matched at high resolution. We let the clinical teams treating the patient know, and they will make arrangements for his sister to travel from New York to London to donate stem cells.
In virology, we provide diagnostic tests that identify a wide range of viral infections that cause disease. The viral load is measured using real-time PCR, and at Barts Health, we test transplant patients weekly in their plasma to measure the amount of CMV DNA present. To do this, we need to extract nucleic acid, so the DNA and RNA from the blood sample, and we do this using an automated extractor. To do this, we need to extract nucleic acid, DNA or RNA, from the blood sample, and we do this using an automated extractor. This one will extract from 24 samples at any one time. The plasma sample is loaded onto the extractor, and the first stage is to lyse the sample using a buffer containing guanidinium thiocyanate, which denatures proteins and releases the nucleic acid into the sample. The enzyme proteinase K also helps this lysis step. Once the sample is lysed, magnetic beads made of silica are added. These beads bind tightly to the nucleic acids and allows the unwanted fraction of the sample to be removed. After washing, a low salt buffer is added that causes the silica beads to release the nucleic acid, and this is then purified and collected ready for any molecular diagnostic test. PCR is the exponential amplification of a double-stranded DNA sequence, and in one reaction, billions of copies of this sequence are generated. Great care has to be taken in making sure that the laboratory environment for molecular diagnostic testing limits the potential problem of PCR contamination that would lead to false positive results. Each laboratory stage of the PCR process is conducted in a separate environment and using a unidirectional workflow, then the possibility of PCR products contaminating the other laboratory environments is minimized. At Barts Health, we have one lab to perform the nucleic acid extractions from the clinical samples, and another lab, the clean room, where the key components of the PCR tests are stored and set up prior to testing. In this lab, the primers, probes, and master mix containing the Taq polymerase enzymes are aliquoted ready to set up the final PCR reactions on the clinical sample. So for CMV viral load PCR, the primers and probes used specifically amplify a DNA sequence of a CMV gene. Other key components include the deoxynucleoside triphosphates that are used in the DNA synthesis of the PCR product. It is very important that the clinical samples and anything post-PCR do not come into the clean room environment. The PCR reactions are set up using liquid handling robots. The use of robots allows this to be done quicker and generally with more accurate pipetting. Patient's nucleic acid extract and the master mix including primers, probe and Taq polymerase are aliquoted. Also included on each run are relevant controls such as positive control, negative control, and for a quantitative test such as CMV viral load testing, a range of standards. The standards are composed of known copy number of the sequence to be amplified by the CMV primers and probe. After all the components of the PCR reactions are pipetted by the robot into the reaction tubes, the disc is then sealed using a heat sealer. Once the PCR reactions have been set up and the tubes sealed, they are taken to the PCR laboratory where all of the PCR thermal cycling is carried out. The PCR tubes are placed in the real-time PCR machine and the run program started. This program consists of an initial extended DNA denaturation step at 95 degrees C, followed by 40 cycles of denaturation and annealing at 95 degrees C and 60 degrees C respectively. The real-time PCR machine will measure the fluorescence in each tube at the annealing temperature stage of each thermal cycle. Most real-time PCR tests utilize a fluorescently labeled hydrolysis probe. In this case, apart from containing a forward and reverse primer specific for the target, there is the inclusion of a probe which is designed like a primer to be complementary to an internal sequence of the PCR product. The probe is synthesized with a fluorescent molecule at the 5' end and a quencher molecule at the 3' end. In this configuration, the probe does not fluoresce, and the energy emitted by the fluorophore is absorbed by the closely positioned quencher molecule, and no fluorescence is detected. The probe will bind the target sequence within the PCR amplicon. As the Taq polymerase enzyme has a 5' to 3' and exonuclease activity, it will degrade the probe as it synthesizes the new strand of DNA. As a result, if the template is present in the PCR reaction, at each thermal cycle, more hydrolysis probe is being degraded. In this state, the energy from the fluorophore is no longer quenched, and the fluorescence from the molecule can be detected. Fluorescence is measured at each cycle, and the PCR amplification will result in the generation of fluorescence following a sigmoidal curve. Once the 40 cycles of thermal cycling has finished, we can analyze the run. In this field, we have the sample numbers and quantitative standards and their tube positions in the real-time PCR machine. A threshold line is plotted on the amplification plot, and any fluorescence line that crosses the threshold is considered to be a positive. That is, there has been a significant amount of fluorescence generated in the PCR reaction above the initial baseline level. When the amplification plot crosses the threshold line, this is noted as the CT, which stands for crossing threshold. Sometimes this is called the CQ for quantification cycle. To make the test quantitative, the quantitative standards are amplified by the same primers and probes as the patient sample. A standard curve is plotted with a line of best fit through a range of standards, and the copy number in the clinical samples can be determined from this plot. Based on the CMV viral load results, then different actions may be taken. If the viral load is high and above a certain level, then the clinicians will be directly informed, and it is likely that the patient will then receive antiviral treatment. For CMV, the first choice of drug is ganciclovir, which inhibits the function of the viral DNA polymerase. The drug can be taken orally as a prodrug, valganciclovir, or may be administered as intravenously as ganciclovir. In patients who receive ganciclovir, because of their CMV viral load results, they continue to be monitored for CMV viral load weekly, and when they have negative CMV PCR results, then the treatment can be stopped. The rationale for either the prophylaxis or preemptive therapy approaches used for CMV in transplant patients is to prevent CMV replication developing to such a high level that it causes CMV disease.
Microbiology is still largely culturally based, requiring the inoculation of large numbers of different plates. You can clearly see the range of different colony types produced and the way the machine has spread them out around the surface of the agar. The introduction of mass spectrometry has revolutionized the way bacteria are identified in the UK and European labs. Bacterial colonies are directly picked off culture plates, mixed with a small amount of matrix and spotted onto a steel plate. The steel plate is inserted into the mass spectrometer, the door closed, and the chamber evacuated to a very high vacuum. A nitrogen laser ionizes the proteins, and the time taken to travel through the system under a high electric current is measured. The protein spectrum is compared to known strains in the database and an identification made. The diagnosis of sepsis has remained essentially unchanged for 50 years or more. 5 to 10 mils of patient blood is taken and injected through the lid of the bottle containing culture medium, which will support the growth of a range of different bacteria. This slide shows what is for now the state-of-the-art approach, the Abbott Iridica system, which can be used to detect bacteria, fungi and viruses directly from patient specimens. A result is obtained in just over 6 hours. The different components of the system are shown in this picture. It's similar to many other techniques that you've heard described, and it's essentially a multiplex PCR reaction, although in this case, instead of sequencing PCR products, they're actually weighed by mass spectrometry. In the first stage, specimens of blood or sterile tissue are placed in a bead beater. The tubes contain sterile glass beads. Once loaded, the machine is switched on, and violent agitation disrupts bacterial and fungal cell walls, as well as any human cells which may contain intracellular bacteria. The air in this room is obtained from outside the laboratory to minimize contamination of the specimen. Following DNA extraction, we move into a separate room with its own air supply, where the next components of the system are placed. The DNA extract is placed in the thermal cycle for a conventional multiplexed PCR with a large range of primers. After thermal cycling, the DNA extract is desalted as the salts in the PCR reaction disrupt the mass spectrometer, so have to be removed. After desalting, products are placed in the mass spectrometer and automatically injected through one of the 2 ports that are shown at the back of the machine and automatically analyzed. Products are identified using the machine's internal database and an identification report produced.
Welcome to the Molecular Hematology Laboratory. We are a team of scientists that perform a diverse range of molecular tests, including the monitoring of chimerism following allogeneic stem cell transplantation. Chimerism studies provide useful information on the level of engraftment. The analysis is performed on both whole blood and lineage-specific white blood cells, notably CD3 positive T cells and CD15 positive myeloid cells. Here we see Shamana loading blood samples onto the Automax Pro, an automated cell sorter. The cells are labelled with lineage-specific antibodies bound to microbeads, which are positively selected by passing through a magnetic column. Prior to DNA extraction from the separated cells, we assess the purity using flow cytometry. DNA is extracted from whole blood and the separated cells using the Maxwell 16 robot. The machine uses a cartridge-based system for extraction. Here, we see Shamana loading a cartridge ready to place on the robot. This machine can extract DNA from up to 16 samples per run, lasting 35 minutes. The purified DNA can be used directly in a variety of downstream applications, including PCR, restriction enzyme digestion, and agarose gel electrophoresis. Chimerism monitoring involves analysis of short tandem repeats, STRs. A PCR is performed using the PowerPlex kit, which amplifies 16 STR loci from both Fred and his sister. The PCR reaction takes 3 hours to complete. The PCR products are run through the shared capillary electrophoresis machine located in the post-PCR suite. The products are separated on the basis of their size. Fred and his sister will have 2 alleles for each STR locus, so there may be up to 4 alleles detected. The results from the capillary electrophoresis are analysed using ChimerMarker. This automated software identifies donor and recipient alleles and determines which loci are informative and can be used in the percentage donor calculations. From this analysis, Fred's transplant has successfully engrafted and is currently showing as 98% donor.
In this virtual visit to the pathology labs at Barts Health, we've given a snapshot of how different disciplines are essential in bringing together a diagnosis of AML in our patient and aiding treatment decisions such as stem cell transplant. With such therapy, there is a risk of infection post-transplant, and we use a range of molecular diagnostics to identify these infections and then treat the patient with drugs targeting specific viruses or bacteria. We also use molecular diagnostics to monitor the success of the transplant to treat AML. We are grateful to the QMUL e-learning production scheme for supporting the development of this video.